Welcome friends and welcome to my channel Sequential Biology. In this video we will discuss the three domains of life. All the organisms in this earth included under these three domains. These three domains are domain bacteria or domain Eubacteria, domain Archaea or Archaebacteria, and the domain Eukarya or Eukaryotes. So each domain contains many kingdoms, and the famous microbiologist Dr. Carl Richard Woese from University of the Illinois proposed three domain system of classification, and each domain contains many kingdoms. Remember it, each domain contains many kingdoms. That means the domain is bigger taxonomic category than the kingdom. In recent years, new discoveries has challenged the most accepted five kingdom system of classification. This five kingdom system of classification was proposed by R.H. Whittaker, but this system of classification was challenged by the three domain system of classification, and this three domain system of classification is more recent because this is based on rRNA. And studies of rRNA showed that all life shares a common ancestor, and there are three major evolutionary lineages called the domain, and domain is the biggest taxonomic group. Then this three domain system of classification is based on rRNA. This is very important, right? And in three domain system of classification, if we consider the kingdom, there you can find 6 kingdom, but in five kingdom system of classification you can find 5 kingdom, but in three domain system of classification you can find 6 kingdom. To rRNA technology, it is revealed that all the organisms belonging onto these three domain are originated from a common ancestor, and which is known as progenote. And from this progenote the three domain arise, and in domain bacteria or the Eubacteria you can find modern bacteria. That means the bacteria or the prokaryotes which are cosmopolitan in distribution, and the representative of this domain bacteria can be found everywhere in our world. But the representative of the domain Archaea or the Archaebacteria are present in extreme environment, and for this reason the representative of this domain Archaebacteria are known as extremophiles. And in domain Eukarya you can find 5 kingdom. The first kingdom is kingdom Protista, and here I have given an example, that is the entamoeba, and these entamoeba are included under kingdom Protista, which is unicellular eukaryotes, and also amoeba are also another important member of kingdom Protista. And also the kingdom Fungi, kingdom Plantae, and the kingdom Animalia, these kingdoms are included under domain Eukarya or the eukaryotes. And apart from this, an interesting group of organisms, that is slime molds, are also included in this domain Eukarya. The slime mold shows peculiar characteristics. They can reproduce by formation of spore-like fungi, but they can move like animals. We will see a brief evolutionary history of this three domain. The Eubacteria are thought to have diverged early from the evolutionary line. This is the evolutionary line. These are the two important evolutionary line, and from this progenote, one line causes the development of domain bacteria or the Eubacteria, but the Eubacteria are thought to have diverged early from the evolutionary line. So from the viruses, the domain bacteria appears, and then the domain Archaea and the domain Eukarya appears, and the domain representative of this domain Archaea and the domain Eukarya shows similar characteristic features, and for this reason the domain Archaea and the domain Eukarya are closely related than domain bacteria. The domain Archaea and the eukaryotes diverged later and are more closely related to each other than either as to Eubacteria. Right? Now these three domains include different kind of organisms. In case of Archaea, and now recently it is called as Archaea, and previously it was called as Archaebacteria. Then the Archaebacteria and the Archaea is similar term, but previously it was called as Archaebacteria. This was the first domain of Carl Woese in three domain system of classification, and now it is called as Archaea. Right? The prokaryotic microbes live in extreme environments such as high temperature, high salt concentration, and the acidic environment are included under this domain Archaea. And next domain is eubacteria. They are the true bacteria and they can found everywhere in our world. But the representative of this domain Archaea or this type of prokaryote are present in extreme environment. And in domain Eukarya, all the eukaryotic organisms, this organism may be unicellular, this may be multicellular, but they are eukaryotes, so they are included under the domain Eukarya. Right? And now we will discuss the characteristic features and the differences among these three domains. Bacteria, Archaea and the Eukarya. The bacteria and the Archaea, they both are prokaryotic and they are single cellular, right? So bacteria are prokaryotic, Archaea are also prokaryotic, and the bacteria are single cellular and the Archaea are also single cellular. But the domain Eukarya, the members of this domain are eukaryotic and the members may be single or multicellular. You know, many algae and the fungi may be single cellular, and also kingdom Protista, which is included under domain Eukarya, are single cellular. And many of the organisms are multicellular. So in this domain, you can find both single and the multicellular organism. In bacteria, just outside of the plasma membrane, there are present cell wall, and this cell wall is made up of peptidoglycan. In case of Archaea, the cell wall also present, but the peptidoglycan absent in their cell wall. And in case of Eukarya, the peptidoglycan absent in the cell wall. And you know, in case of fungi and the plantae, their body or their cell is covered by cell wall, and in fungal cell wall, the main component is chitin, and in plant cell wall, the main component is cellulose. That is, the plant cell wall is cellulosic. So in both these two important kingdom under the domain Eukarya, their cell wall is not made up of peptidoglycan. In fungi, the cell wall component is chitin, and in plantae, the cell wall component is cellulose, or there may be present hemicellulose, pectin. So different kind of materials can be found in cell wall of plant, but the peptidoglycan is absent. And next, cell membrane. And you know, the cell membrane is made up of lipid, and the lipid is made up of fatty acid and the glycerol, but the lipid structure are different in different type of domain. And in case of bacteria, the fatty acid are unbranched. In case of Archaea, the fatty acid are branched in their plasma membrane, and also in case of Eukarya, there you can find unbranched fatty acid, and the fatty acid are joined with glycerol, and this will form the lipid. But the bond which causes joining of this fatty acid and glycerol are different in different domain. In case of bacteria, the glycerol attached with fatty acid by ester bond, and in case of Archaea, the glycerol attached with fatty acid by ether bond, and in case of Eukarya, the fatty acid attached with glycerol by ester bond like bacteria. Then the cell membrane is made up of lipid, and in this lipid, the unbranched fatty acid can be found. And in case of Archaea, the cell membrane also made up of lipid, but the branched fatty acid can be found. These are the branched fatty acid. And also in Eukarya, the cell membrane is made up of lipid or the phospholipid, but in this lipid, the unbranched fatty acid can be found. And in this fatty acid, these are the phospholipid actually, you can say, and they are present a polar head group and the non-polar hydrophobic tail group are present, the two tail group present in fatty acid. And usually the main lipid component of the plasma membrane is fatty acid in all these domain, right? And in case of bacteria, the fatty acid attached with the glycerol by ester bond, so this is the ester bond. And in case of Archaea, the fatty acid attached with the glycerol by ether bond, and in case of Eukarya, the fatty acid also attached with the glycerol by ester bond. These are similar with bacteria. In bacteria and the Archaea, no membrane bound organelle can be found. Since they are prokaryotes, so obviously there will be no membrane bound organelle like mitochondria, plastid, Golgi body. So this type of membrane bound organelle cannot seen in these two domain, but the membrane bound organelle can be found in domain Eukarya. Since they are eukaryotes, obviously there will be present membrane bound organelle. And since bacteria and the Archaea are prokaryotes, so no nuclear envelope can be found in their nucleus. But the chromosome can be different in different type of domain, and the bacterial chromosome is only made up of DNA, so their chromosome is single circular double-stranded DNA molecule, and in their chromosome no histone protein can be found. So the DNA without the histone protein and this single circular DNA molecule can form the chromosome in case of domain bacteria or Eubacteria. And in case of domain Archaea, the DNA is also circular like bacteria, but histone like protein can be found. This is not true histone, but histone like protein can be found in domain Archaea. In case of Eukarya, the DNA is linear. So this is the linear DNA, and this differ from other two domain, right? In bacteria and Archaea, the DNA is circular, but in case of Eukarya, the DNA is linear structure, and also in their chromosome histone protein can be found. This is a very important characteristic features of eukaryotes. And if we consider about their genome structure, the introns are absent in case of bacteria. Introns are inactive gene, they cannot synthesize protein, so they form the junk chromosome or the junk DNA, and this junk DNA are absent in case of bacteria or Eubacteria. But in case of Archaea, introns present in some genes. This is very important. Introns present in some genes in case of Archaebacteria, and intron present in eukaryotes. And for this reason, they form a huge inactive genome. And this huge inactive genome or the intron are present in Eukarya. But in case of Archaea, in some gene, you can find intron. And next portion is also very important, and you know the protein is made up of different type of amino acid. If we consider their protein structure, and the protein is made up of many amino acid, and this amino acid are linked by peptide bond. But in case of bacteria, the first amino acid is formilated methionine. The amino acid that initiate the protein synthesis is formilated methionine. But in case of Archaea, the amino acid that initiate protein synthesis is only methionine. But in case of Eukarya, the amino acid that initiate protein synthesis is methionine, right? This is also a very important features. Now antibiotic, and when we study the bacteria, the antibiotic is very important because this inhibit the growth of bacteria. Then the response to antibiotic streptomycin and the chloramphenicol can be found in case of bacteria, but Archaea and the Eukarya do not respond against this antibiotic. And also their growth will be inhibited in presence of different antibiotic, but in case of Archaea their growth will not be inhibited, and also in case of Eukarya their growth also not be inhibited in presence of antibiotic. That means in Archaea and Eukarya, antibiotic do not affect their life cycle, right? So these are the basic differences among these three domain and their characteristic features, and hope you have understand very clearly about these three domain. And in next video we will discuss different group of Archaeabacteria. So thank you, and that's it.